who had inspired the juniors a lot to the hard work and to go beyond the crowd. And that's exactly what he has achieved in his own life. About that, our Honorable Principal Madam is going to shed light upon in details. Now, to have a hint, will be this much will be enough to tell that for past 10 years, he is continuously working on insect-related zoological work, spanning from taxonomy to ecology, with the Zoological Survey of India. And we are here actually to share some of his well-earned experiences among us. Now, I shall request all the participants to follow all the rules and given by our technical team. They are working very hard to make all these webinar go successful. So please cooperate to them. And during question answer session, we will accept our questions only from the that has been typed in the chat box. So try to type your question in the chat box, because turning on all the microphones together will make it a mess. So also the host is Dr. Moirakhi Nag. I mean, she has been there? Okay. So I'm requesting Dr. Moirakhi Nag, who is also the joint convener of this event. So thank you all.
Moirakhi ma'am, unmute yourself.
I think she has been mute. Someone.
Dr. Moirakhi Nag?
Am I audible?
Yeah.
Good afternoon. I'm Moirakhi Nag. Welcome you once again to the webinar and hope that you will have a great time ahead. Now I'm honored to invite our principal madam, Dr. Shoma Roy, to deliver a welcome speech. Madam, over to you.
Good afternoon, everybody. Respected resource person, Dr. Devanshu Gupta, scientist, Zoological Survey of India, respected teachers of different colleges, scholars, and other virtual audience. It is a privilege to extend my sincerest welcome to each of you to be in this webinar on Insect Diversity of India, Collection, Preservation and Identification. Before the outbreak of COVID-19, our teaching and learning was mostly dependent on chalk and talk method, that is the classroom teaching. In last 3.5 months, we remained at home except some urgent need. Both human lives and livelihoods are being upset. All of us remain in fear. Still, we are trying to continue our activities in this new normal period. Both teachers and students are being accustomed with the online teaching learning method. We are joining in webinar, video conference, online courses, etc. But one positive side we should not ignore about this virtual platform, that it has been possible to hear speeches from those eminent persons who may not be physically appear in our college in a seminar like today's resource person. I extend my heartfelt gratitude and thank you, sir, for having you in this webinar for your valuable lecture. I hope a very fruitful and enlightening discussion in this webinar, which will enrich our virtual audience. I think our students will be benefited much from your talk. I wish a grand success of this seminar. At last, I wish good health with sound mind for all of us in this COVID-19 pandemic. Thank you all for joining in this webinar. Thank you. Namaskar.
Thank you, madam. Thank.
Now, our IPSC coordinator will speak about the webinar.
Thank you, ma'am. Thank you, everybody. Good afternoon, everybody. On behalf of the principal of Sirampur Girls College, Dr. Shoma Roy, IQAC members, webinar committee, the Department of Zoology, and all other professors and staff of this college, I, Dr. Shayanti Mukhopadhyay Talukdar, IQAC coordinator of Sirampur Girls College, welcome you all to this webinar. The Department of Zoology, in collaboration with IQAC, has organized a national level webinar today. The topic of the webinar is Insect Diversity of India, Collection, Preservation and Identification. We have with us the renowned scholar and a scientist, Dr. Devanshu Gupta, Scientist C, Zoological Survey of India. I cordially welcome you, sir, to this webinar. Extremely thankful to you because you are going to give us your precious time. The world in the grasp of COVID-19 pandemic has been threatened every single moment for the last few months with the annihilation of life from Earth, putting a big question to the continuation of human existence itself. But this is limiting to human life only. In the midst of it all, we humans have almost forgotten the fear factor gripping us every moment, that there is still life on Earth. There is life in the form of other animals, birds, beasts, and insects. That there is nature that needs our attention, our love, which has somewhat been ignored. Therefore, the topic of today's webinar, the insect diversity, is extremely important, not only from the scientific zoological point of view, but also for sheer reverence that life within quote demands. Chris Peckham, TV presenter, naturalist, and founder of Wild Justice, says, quote, nature is our life support system, and without it, our lives on this earth would be impossible and unimaginable. Nature matters to me, and it should matter to you. We need to put more value on our natural assets and stop destroying our special planet, unquote. If we are truly to live within the sustainable bounds of our extraordinary planet and leave the space of nature that it so desperately needs, we have to step back and be more considerate about the way we treat our world. Nature provides us with the food we eat, air we breathe, and the water we drink. We depend on the nature to grow our crops, to source our medicines, to house us, and to clothe us. Therefore, when we destroy nature, we are destroying the very essentials on which we all depend. However, this is the anthropocentric view of nature. Norwegian philosopher Arne Naess had coined the term deep ecology. He was an intellectual and an inspirational figure within the environmental framework or environmental movement of late 20th century. The term deep ecology emerged from the rejection of anthropocentrism or human-centeredness. Deep ecology proclaims that all living things have intrinsic value, first. Second, diversity of life forms contribute to the realization of these values. And thirdly, humans have no right to reduce the richness and diversity of this except to satisfy the vital human needs. We have never had a better chance to make a greener world. COVID-19 has delivered unusual environmental benefits, cleaner air, lower carbon emissions, a respite for wildlife. Now, the big question is, can we really help environment survive? Whether we can capitalize on this movement? However, there is still some time to protect what is left and to start restoring nature. But to do that, we must radically change the way we live, and each of us has a role to bring about this transformational change. Insects are the vital part of this nature. They are the most ubiquitous, successful, and dominant forms in the whole of animal kingdom and are remarkably adaptable to a variety of environments. India, with 2% of global space, is one of 17 mega diversity nations in the world in terms of insect diversity, with a very rich and diverse insect fauna. Collection, identification, and characterization of insects and mites that impact Indian agriculture and biosecurity include pests of crops, alien invasive species, and beneficial insects, including insect predators and parasitoids that can be used for biological pest control, pollinators, insects involved in soil building and nutrient cycling. And again, some can be valuable sources of chemicals and medicines. Insect biodiversity has a significant role in the maintenance of the ecosystem. So there is increasing need for taxonomic information and services in our society, particularly for biodiversity assessment for attaining an environmentally sustainable future. Biodiversity is not static. It is a system in constant evolution from a species as well as from an individual organism point of view. The Zoological Survey of India is an important institution in the Department of Environment, Forests and Wildlife, Government of India. It endeavors to impart knowledge and expertise acquired by it to the research workers and scientists working in various medical, veterinary, agriculture, forestry department, and also universities, and also to amateur collectors and naturalists. I must say that we are extremely privileged that we have among us an esteemed scientist, Dr. Debanshu Gupta from the Zoological Survey of India. He will be delivering a lecture and imparting knowledge on this very important aspect of biodiversity or insect diversity of India, which I'm sure will enrich the students, the research scholars, as well as the teachers. Sir, extremely grateful to you for your presence here. I thank the audience for your patient hearing. সবাইকে জানাই অগ্রিম শুভেচ্ছা, অভিনন্দন। সুস্থ থাকবেন, ভালো থাকবেন, আপনাদের আন্তরিক ধন্যবাদ। Please stay safe. Thank you all once more. Now over to the
Thank you,
Department
madam.
of Zoology.
Thank you, madam. Thank you
you
for
so
the
much.
beautiful narration.
I'm privileged. Thank you.
Now, without further ado, we will turn the time over to the speaker, Dr. Debanshu Gupta. Sir.
Hello. Am I audible? Hello.
Yes.
Yes, sir, you're audible.
Good afternoon, everyone. Respected Madam Dr. Soma Roy, Principal, Srirampur College. Thank you so much, Madam, for inviting me for this to give a lecture on insects of India, collection, preservation, and identification. Madam Dr. Sayanti Mukhopadhyay, you know many things about ZSI, insects, and you have briefed many things. All the things, most of the things you have briefed about, and that will be easy for me to go for
I try to do a little bit of research, sir. Thank you. I do not know much. I really do not know much anything.
But it will really help me in the lecture, because the participants will be having now some background about insects, what they do, and also about ZSI. Madam Principal, Madam, I humbly thank you from the department, my department, Zoological Survey of India, and from my own behalf. Thank you for inviting me in the conference and all the participants for joining the webinar. And my friend, Dr. Ananya Ali, thank you so much. Dr. Mayur Kashi Nag, thank you. Please wait for 1 minute. I am sharing my presentation. As you all know, first I will brief about myself. Hello. I would like to brief about myself. I am Dr. Devanshu. I am scientist at Zoological Survey of India, and I am working on insects since 10 years. I did my PhD on dung beetles, and then from Jabalpur Zoological Survey of India, I studied dung beetles from Madhya Pradesh and Chhattisgarh. Then I came to beautiful city of Kolkata in 2015. Then onwards, I am working on different aspects of insects, and especially I am dealing with the beetles. Now I am in charge of Coleoptera in the Zoological Survey of India. I would like to brief something. My presentation is loading. Please wait for some time. Zoological Survey of India, you all know, it is the only organization in India which is dealing with the faunal diversity. Faunal diversity from, as I came to know that most of the students are of BSc, MSc, and many of the research scholars are there. Faunal diversity from protozoa to mammalia. Hello. We in Zoological Survey of India deal with the faunal diversity of from protozoa to mammalia. Okay. कनेक्ट नहीं हो रहा था, यहाँ से ज्वाइन करिए, यहाँ से. Zoological Survey of India was कुछ प्रेजेंटेशन में अपने में लगा दो. Please for 2 minutes. इंटरनेट तो आ रहा है, इंटरनेट आ रहा है. अपने है ना मेरी ईमेल आईडी. The Zoological Survey of India was established in 1916 with its headquarters in Calcutta, and it's dealing with mostly fauna from protozoa to mammalia. Attach हो रहा है क्या?
Any problem is there?
Please wait. Something disconnectivity happened here.
Okay.
Please wait, Dr. Ananya. Please wait for 2 minutes.
Okay.
Please. Pen drive presentation.
Sorry for little inconvenience. I think there are some technical difficulties during uploading that presentation. Just keep patience, he is coming.
Hello.
Yes, sir.
Hello.
Please tell.
I am extremely
Yes, sir.
sorry for the inconvenience. I'm extremely
No,
sorry.
no worries. Please continue.
Please, this Zoological Survey of India, I was telling about that it was established in day back in 1916. It has hit its headquarters in Kolkata and 16 regional stations in different areas. Like in West Bengal, we have 3 regional stations. One in Sundarban Canning, and one is in Digha, and one is in Kolkata. It is actually Zoological Survey of India deals with the faunal diversity from protozoa to mammalia. We actually survey, inventorize, and collect and identify different group of organisms ranging from protozoa to mammalia. We do in Coelenterata also, we do in Porifera, we do in Platyhelminthes and insects, mammals, reptiles, and we in ZSI, apart from taxonomy, we also have started working on climate change. Impact of climate change on especially of insects, Lepidopteran insects, on their distribution shift and their status. The first director of the organization was Dr. T.N. Anandale. It was in 1916, and till now we have 17 director is working. He is Dr. Kailash Chandra, one of the renowned entomologists in India and the world, and he's heading the department right now. Zoological Survey of India, as you all know, it is the custodian of National Zoological Collection. We most of you people must be knowing that we in Zoological Survey of India we have repository, means repository of specimens, and Government of India recognized Zoological Survey of India as the custodian for all the national zoological collection. And after Biodiversity Act in 2002, it was restricted by some 11 institutes like ZSI, ESI, IRI, NBIR, NBP, Fish Genetic Bureau, Lucknow, FRI. These organizations were designated by the Ministry of Environment, Forest and Climate Change for maintaining the collection of different group of animals. We are having not only and I will be happy to share with you that in ZSI we have more than 55 lakhs of specimens. 55 lakhs of specimens in different stations, regional stations, and headquarters as well. So since its inception in 1916, ZSI scientist has described more than 5,000 species new to science. And you will be surprised to know that our country is having more than 1 lakh species, animal species in India. Current number is 1,01,684 animal species. Out of them, 3,500 are protozoans, and rest are Poriferans, Coelentrates, Platyhelminthes, and other groups. ZSI also deals with the fauna of states and union territories. As we all know, we have political boundaries also, and so we do document the fauna of different states. Like West Bengal, it was done in way back in 1995 nearby, and 10-12 volumes have been published on the West Bengal fauna. You can all go and see these West Bengal state fauna series in faunaofindia.nic.in. There is a website. All the publications of ZSI, which include 1,550 books, 10,500 papers, they are all freely accessible at faunaofindia.nic.in. And moreover, ZSI also document the faunal diversity in different conservation areas, like protected areas, our national parks, wildlife sanctuaries, conservation reserves, community reserves. And we have also started DNA barcoding, and one of the leading in DNA barcoding in India. Till now, we have submitted 4,000 DNA barcode sequences in whole database. Then now coming to my main topic, as Madam Talukdar Madam has clearly told about the value, about the why insects are important to us. But for knowing all those things, we need to understand what actually are insects and what their diversity in India are the world, and how many types of insects. And we deal this science in that is called taxonomy. Taxonomy, as everybody, you people must be knowing that taxonomy is the science of naming, classification, and identification. First, we identify a particular species, then we classify it, and then we name it. That is the field of taxonomy. And these insects are belong to class phylum Arthropoda. And the main feature you can distinguish with any other animal, any animal which is having 6 legs and jointed appendages is an insect. And they belong to phylum Arthropoda, and economically they are very means in terms of you see, as Madam told, that honeybees and all those things, they are both beneficial and harmful. Beneficial in the sense, I will say, you must have heard about sericulture, lac culture, apiculture. Now, in apiculture, the bees are important for giving honey, wax. Not only this, bees are very important. They are pollinators of crops. And in recent this IPBES, it has been estimated that about 85% of cross-pollination has been done by only bees. And suppose if bees are absent, how come this food will be provided? And they are very important for our food security also. Like we have very huge population in India, 135 crore people, and all over the world. So the bees, if bees get extinct, or the population of bees gets lowered, then ultimately the food productivity will be lowering because of this pollination issues. And in case, and many of the insects are agriculture pests also. Must be they are destroying crops and forestry. In forest also many of the insects are pests, like you must have heard about Hoplocerambyx, spiny cornix, saw hardwood borer, teak defoliator, and many other insects. And these are few examples I am telling to tell you about the importance. And for studying taxonomy of a particular group, you must know a set of characters. We study characters. So insect body is basically divided into 3 parts: head, thorax, and abdomen. We all know about this, and all the this bilateral symmetry is there. First thing is there. Then head is having different appendages, ocelli are there, antennae are there, mandibles are there, clypeus is there, labrum is there, maxillae, labium. These all terminology we need to know so as to classify insects or animals in different groups. All the groups are having. Okay. Then based on this, 2 pairs of wings are there, 3 pairs of legs, and different legs are divided into coxa, trochanter, femur, tibia, and tarsus. Okay. Then different types of mouth parts. You must be having, must have seen some beetles chewing the leaves, and butterflies taking juices from these flowers. So this they evolved through co-evolution. As the angiosperms evolved and as the insects evolved, they evolved together, both of them, and that's why they are having different types of mouth parts also. Like beetles are having biting and chewing type of mouth parts. They, in that case, mandibles are very strong. Insect mouth parts are having different parts, like 1 or 2 pair of mandible, 2 pair of maxillae, 1 labium, and 1 labrum, and hypopharynx also. And these, this is basic. This left side, this is basic structure, which is modified in, like you see houseflies, when it sits, certain that humid thing comes. means they secrete some enzymes, then they first secrete lysozymes, then they suck the things. Okay, the this butterflies and moths, the mouth parts are modified into proboscis, because they need to reach to the bottom of the flower for sucking the nectar. Okay. In bugs also, bugs are mostly having sucking type of mouth parts, so they also pierce inside the leaves and then suck the flower. Beetles are having mandibles. So as something about their life cycle also, in case of insects, the life there are four life stages major: egg, larva, pupa, and adult. But in it evolved, means in case of certain groups, primitive groups, the life cycle is hemimetabolous. Three types of life cycles are there: hemimetabolous, holometabolous, and parometabolous. In hemimetabolous, like grasshoppers, bugs, you must have been seen the juvenile ones, like cockroaches in the houses. You must have seen they resemble with the adult ones. So this is hemimetabolous. The egg stage is followed by nymphal stage. Nymphal stage is non-reproductive stage, and the adult stage. Adult stage will be having well-developed wings, and then they will lay eggs and die. But in case of holometabolous, you take the example of butterflies. The adult butterfly and the juvenile, larva, do not resemble each other. Means they both are different. And larva is a feeding stage, and it will destroy all the leaves, flowers, whatever they will get, they will eat. And then the pupal stage will come. Pupal stage is a resting stage, and from there the adult will emerge. In case of this odonates, certain special type of metabolism is there, that is parometabolism. Insects are habitat specialists, which means you check for any plant like oak tree. In every particular part you will find insect, and they are specialists. In roots you will find different type of insects, like termites you will find, beetles you will find. In leaves you will find different type of insect. In canopy, upper surface, upper part of tree, you will find different type of insect. Very specific niche insects are having. So when you go in the field, you just look what you want to see. You search everything, you will find insects everywhere. Means in the bark, inside the bark, on the bark, everything, and this debris everywhere you will, and they are specific also. The question now comes: how many species of insects are there? We actually live in the world of insects. Means we human beings are most populated, and we are destroying habitat and everything. We are changing according to our needs. But out of 18 lakh animal species so far described from the world, 10,53,000 species are of insects. You see 66%, nearly 67% species are of insects only. And that if you come to arthropod, it will go beyond 78-79%. But this 10,53,000 species which we know through taxonomy, people have described the species, people have studied them, but there are many species we do not know where are they. And the taxonomists in the world are continuously describing the species. You must be hearing in the news that some new species is described, some new genus is discovered. So the question is, we now have 10,53,000 something species, but estimates suggest that the total number of species on this planet is nearly 55 lakhs. Means 45 lakh species are yet to be discovered. We have only discovered 10,53,000 species. And out of that, beetles is the most diverse and abundant order, and described beetle species is nearly 3,86,000. So you see, in broad perspective, that insects are everywhere, and lots of work need to be done in case of insect discoveries. Okay, so 10,63,000 hexapods are known. And in India, as I previously told you, that the total faunal diversity is nearly 1,01,681 species, and out of them, you see 65,466 species are of insects only. Means in mammals you have only 429 species, in birds you have 1340 species, in reptiles you have nearly 600 species, in amphibians you have 438 species, but you see the group insects, it is having 65,476 species only from our country, and many of the area is still uncovered. For those entomologists, I would like to say that Zhang in 2013 presented a classification of animal biodiversity, and especially they briefed about the classification of insects, and in them 39 insect orders were included. Out of them, I will just first give you the brief idea about insect, then we'll tell you how to go for their collection and their preservation, and then for identification, what you will do in the field and everything. 26 orders are present in India. Okay, so eight major orders. I will say the Lepidoptera. Lepidoptera includes butterflies and moths. Coleoptera, beetles. Orthoptera, it includes grasshopper, crickets, these Diptera, flies. Hemiptera, bugs. Odonates, this dragonfly, damselfly. Hymenoptera, bees, wasps, and bees and wasps, thrips and Thysanoptera. These 8 orders means out of 26 orders in India, these 8 orders constitute 94% of the diversity. Other orders are very less represented, are very less species rich. Okay. Then this is recently in 2009, Cullen published the phylogenetic classification, Columbola. Then it is from A. Terrigota to Terrigot, how the evolution of the insects have taken place. And I told you that 65,476 species of insects are known of. 62 species live only in aquatic ecosystem. So you see, most of the species are, and these aquatic species are very important as per pollution and all these things are related, like ephemeropterans. You must be having some idea about these mayflies. They evolve when raining is there, and these dragonflies, they are susceptible to the change in the environment. Like certain species will be found in contaminated water, certain species will be found in without unpolluted water. So these aquatic species, aquatic insect species are very important in terms of analyzing the status of pollution in the environment. I will say that these ephemeropterans, odonates like dragonfly, damselflies, plecopterans, stoneflies, then certain aquatic bugs, and this trichopterans, caddisfly, they are very important in terms of analyzing the pollutant in water. Okay. Now there is one paper, that is Insect Fauna of States and Union Territories. It was published in 2013. It is a baseline till now in 2011 and 13 subsequently. It was revised in 13. Then it is the baseline information for insects of India. You can go and search in Google. You will get this paper as PDF, and this is very useful for MSc and PhD and the teachers for the teaching community for having an idea about what about insects and what is their status, especially in India. These are the number of insects reported from different states, and this list, this is just an idea to show you what is the status, but this is continuously changing because of the discovery of the species, because new species are being described on daily basis. In every journal you search, like Zootaxa, Zookeys, Coleoptera bulletin, all those journals, you will find every day new species are being described every day, and these numbers are continuously changing. You see in West Bengal, nearly 5,818 species of insects are known. You see why this this is very rich in terms of other group. Why this area is so much rich? The question is, it's not like West Bengal is having undoubtedly the mountains, the mangroves, the Gangetic plains. Every particular habitat is here, but the main thing is why this area is having is because most of the surveys were done in Sikkim, Darjeeling area. That's why this region is having more number of species, like 5818. The most number of species of insects known from India is from West Bengal only. Okay, and our Zoological Survey of India is also situated in Kolkata. That also helped a lot in documenting the West Bengal insect as well as faunal diversity. Okay, so as I previously told that there are 26 insect orders and in subphylum Hexapoda. And now this I will tell four classes are there. One by one I will tell you about each class. These are certain groups, Collembolans. These are called springtails. Okay, they are mostly found in underneath the soil, means debris and all this. And ecosystem, it is very important, and you can identify these springtails by structure underneath his first abdominal segments. You see these long tails. There are 342 species right now known from India. The other class in insects are proturans. They are telson tails, commonly called as. They are also they are mostly found in soil, and these proturans you can identify with no antennae and no eye, no antennae or eyes. There is no antennae and eyes in proturans. Okay, only 20 species of this group is known from India. You see, West Bengal is having only one species, and rest other species, other 20 species also are present reported from Kerala. The entire India is blank. This does not mean that proturans are not reported from any other state, but they are present in other states, but they have not been studied. So I request all of you to go and collect so that these blank states may be filled, and these things our knowledge of diversity of insect may enhance. Okay. The other group is class Diplura. It can be they are soft-bodied wingless insect. Mostly they are these Collembola, Protura, Diplura. They are wingless, and they can be identified by the cerci, last two one pair of.
Excuse me, sir. So wingless means aterigot?
Yeah.
Excuse me. Actually, so aterigots. Okay.
Yes. Aterigots. Means aterigot, the wing without wings, na? Yes. The 18 species of this particular group is known from India, and in this case also the most of the states we do not have any record of diplurans. These two groups, you see, Protura and Diplura. These two groups are mostly untouched, and anybody interested can go in which state they are. They can go and collect, and in ZSI we can help you in identification of those species. You can send the samples. We can help you in the identification or the photograph. Then comes to the order Insecta. Actually, insects come under subphylum, under Arthropoda, phylum Arthropoda, subphylum Hexapoda. And in Hexapoda, there are 4 classes. I already told that one is class Collembola, second one is class Protura, third is class Diplura, then the 4th class is Insecta. Okay, these are 4 classes come under subphylum Hexapoda. Previously, the classification was different. All these other classes, Protura, Diplura, and Collembola, were placed under Insecta itself. But after the phylogenetic and molecular analysis, the classification is now changed. Okay, and I already told you 65,094 species are reported from India, out of 10,53,000 species from the world, and and these are the status of beetles, Lepidoptera, Hymenoptera, all these groups. 8 major groups are very important. Okay, and out of them you see 65,000, 22,000 are of beetles only in India. This data is of India only. Okay. Now I will come to one by one to each order. Class Insecta is having 26 orders. Okay, so 26 orders out of 39 orders known from the world. First is Archaeognatha. These are bristle tails. These are also apterygotes, and you can identify them by a long tail, 3 long tails, and with a median filament. Okay, and from West Bengal there is no record of this particular group, and most of the record is of Himalayan region. Okay, and I will be happy to share with you. I authored a book on faunal diversity of Indian Himalaya, and you will be surprised to know that more than 30,377 species of animals are known from Himalayan region itself, and out of them 24,000 species are of insects. So you suppose how diverse is our Himalayan region is, that Western Himalaya, Eastern Himalaya, and this Northwest Himalaya, especially in the states of Himachal Pradesh, Uttarakhand, Sikkim. Sikkim Darjeeling is the most diverse region in India as far as diversity of animals is concerned. I am dealing with the insect diversity of India in checklisting and all those things. So you can say that Sikkim Darjeeling, most of the species have been described because the Britishers were there and they collected the specimens and described them. And you will be surprised to know that 15,000 species of different group of animals are known only from the Sikkim and Darjeeling area. Okay, then we'll go to Gigantoma silverfish. Everybody must have seen in their books, and very few 29 species of this group are known, and they are pest of different household and library thing, and 23 species of this group are endemic. Okay, then mayflies. I told in aquatic diversity that they look like mosquitoes, but they are not mosquitoes. You can differentiate them with these 2 tails. You must have seen in your houses that there are certain when light is on, there are certain group of insects will come and they look like mosquitoes, but they are not mosquitoes. You can differentiate them with these 2 protruding structures, oviparous. And 146 species of Epimeropterans are known from India, and this is very well worked out group. Dr. Subramaniam, Dr. K. Subramaniam from Chennai is working, and has described many species also on this group. Okay, and they are very good bioindicator of environmental pollution, especially in aquatic environment like water and all. And they are all aquatic insects. You can put them. Okay, then odonates and dragon and damselflies. Everybody knows 14488 species of this group are known from India, and out of them 186 are endemic. Okay, so these are certain damselflies from Himalayan region. Then all dragon and damselflies. Then orthopterans. Orthopteran, they are mostly, orthopterans includes these grasshoppers. You can see grasshoppers, crickets, mole crickets, and all these. And mostly this group is pest. You must have seen our locust in Rajasthan, Uttar Pradesh, and Madhya Pradesh recently. Locust swarming in these areas are heavily devastated our agriculture crops, and government has to take the decision to spray pesticide for controlling these locusts, and that is going to reduce our agriculture productivity in those states only. And this group is very well worked out in India, and especially in ZSI Kolkata. Very good work has been done, and most of the states we have a very good diversity in case of orthopterans. You must be surprised to know that 1,076 species of orthopterans, which include crickets. Like in third image you can see it is a cricket. Then Schizodactylus monstrosus is a very important species, only present in one of the representative in India. It is distributed in central India. Then this is the book published. You can entire Arthroptera information you can get from this book. It is available. You can note one thing, that there is one website, that www.faunaofindia.nic.in. You can find very good information on insects and any other group also. Platyhelminthes, Cnidaria, Mammalia, many good handbooks will be there on this website. You can download them freely as PDF. Okay, then stick insects, then that includes, everybody knows about them, and there are 144 species of stick insects known from India, and you see many of the Gangetic Plain area like Uttar Pradesh, and Himalayan state Uttarakhand, and all these areas like Rajasthan, arid area, semi-arid area, arid and semi-arid both. No species is reported from these areas. So they are present, but they are not reported because work is less, and most of the species are reported from northeastern region. Then Emboptera, very rare group, and in identification itself, they are found in soil mostly. And one of my colleague, Prasanjit, is working on this group. Dr. Prasanjit is assistant professor in West Bengal. He is working. 31 species of this particular group is present in India. Okay, and many of the states, you see Chhattisgarh, no record is there. Odisha, no record is there. Uttar Pradesh, no record is there. Arunachal Pradesh, no record is there. Arunachal Pradesh, I will suggest the people who are working on taxonomy. Arunachal Pradesh is one of the very important hotspot for biodiversity, and you go and search for insects, you will find many new species in this state, especially Arunachal Pradesh, I will say. And stoneflies, we recently published a catalog of stoneflies from India. 128 species are known of this group. They are aquatic environment they are found, and most of the species of Plecopterans are found in high altitude areas. High altitude area, 89 species. Out of 128 species, 89 species are known from the high altitude areas of Himalayas. Especially this Assam area, this Sikkim, Darjeeling, and you see 20 species from Himachal Pradesh and 15 from Jammu and Kashmir. Okay. Then earwigs, they are also very important. They are found in soil mostly, and can be easily identified by forceps like Wolfster, and very well bird group. We have Fauna of India for Dermaptera, 3 volumes of Fauna of India. You can identify all the Dermaptera of India through those volumes that is present in Fauna of India website, as I previously told. Then the mantids, Dr. T.N. Mukherjee of Calcutta University, he worked a lot on mantids. You must be, he is a renowned taxonomist of mantid in India. And he in 1995 published one paper on this mantids of India and oriental insect. That is still the most important paper for identifying mantids in India. 174 still is working and contributing in mantid taxonomy. 174 species are known from India of mantids. Okay, and it is well worked up by, and the credit goes to Dr. Mukherjee. Then cockroaches. Everybody knows we are 186 species of. We must not be knowing that how many. We know only Planeta americana, Bleta oriental is only 2-3 species. But you see 186 species of cockroaches are known, and you can go for one recently published Zootaxa paper, that is annotated checklist of cockroaches of India, that is available in Zootaxa. You can download through ResearchGate if you will see. Okay, then Platodia. Previously, these cockroaches and these termites. Termite was a separate order. Isoptera was a separate order, but in recently this became a suborder of Platodia, and this group came under Platodia. Okay, now Platodia includes cockroaches and termites both, and 289 species of termites are known. Okay, they are polymorphic insects, and lots of things are to be told, but because of time extent, I will stick to something. Then book lice, 122 species of book lice are known from India, very important group. Then lice, this over 446 species of lice are known. They are ectoparasite of birds and mammals. Okay, this Then thrips. Thrips is also very important group in case of agriculture crops. They are invasive alien species that Madam Talukdar briefed about invasive alien insects and all those things. So some of the thrips like Frankinella, and these are invasive alien species also. And most of the thrips are pest of various crops. Okay, 739 species of we in ZSI has a very good laboratory for thrips, and Dr. Ananta Krishnan is the pioneer of thrips taxonomy. He was in Loyola College, Chennai. Okay. Then bugs, Hemiptera, it's one of the important group. They are also mostly pests, but certain groups are predators also. Some reddivids are predators. 6,479 species of bugs are known from our country. And it is the fifth largest group of insects after Coleoptera, Lepidoptera, Hymenoptera, and Piptera. Okay. Then Hymenoptera. Hymenoptera, they are very important group in terms of both as pollinators, bees. Like I told you previously that bees are pollinating different crops and all those things. Then they are also parasitoids also. Many of the agriculture pests, especially Lepidopterus pests, these trichogrammids parasitoids, they control these pests, biologically control. Many of these species are biological control agents also. Wasps are there, and you will be surprised to know how many. We mostly know about bees for only 4 species: Apis dorsata, Apis sirena, Apis florea, Apis mellifera. But you will be surprised that nearly 817 species of bees are known from India. 817, and most of them, 367 species, are known from our Himalayan region. Okay, these are certain Himalayan species. This is Apis endreniformis. This is our Indian endemic. Endreniformis is present only in India. Apis sirena. This Bombus. Bombus is very important group, and they are very susceptible to climate change. 50 species of bumblebees. Hello. 50 species of bumblebees are known from India. So, the climate change is affecting all those bees. And we know only some 4 or 5 species of bees. Lots of bee species, and they are very important for. And the assessment except these Apis bees, we do not know about the pollination for other bees. But in Calcutta University, Dr. Professor Parthiba Vasu is doing some very good pollination work on pollination. Then certain Strepsiptera, 21 species are known. These groups we do not see in the field nowadays. It's very less in number. Very few experts can identify these groups in these Strepsiptera. This Coleoptera, this is I am working on beetles. 22,299 species of beetles are known. They can be identified by their winged elytron covering in a straight line, making a straight line. And among Coleoptera, 6-5 families. Though from India, 114 families are known: Staphylinnidae, Chrysomelidae, Carabidae. Staphylinnidae is rope beetles, Chrysomelidae is the leaf beetles, Carabidae ground beetles, Porpolinidae, Scarabidae, Cerambicidae, Lateridae, and Tenebromidae. These 6-7 families constitute majority number of species. Okay, so these are beetles are mostly you can see they are pests as well as predators also, and biological important agents also. Some of the have heard about ladybird beetles, family Coccinellidae. So they are very important group of animals in insects which control other groups like bugs. They eat aphids and other groups, and they control them biologically without any use of pesticide. These lady beetles and ground beetles also some predator species. Then these are some pioneer works through which you can start.
Excuse me, sir.
Please.
Excuse me, I just have one from this graph. So somehow this graph is trying to imply that India has a lower diversity of beetles than the rest of the world?
No. In India, 22,000 species are known. Okay, and from the world 386,000
Okay.
species are known. You see the diversity. Okay, so diversity of
Okay.
India in case of beetles is very high. This graph is showing the diversity in different families.
Yeah, actually, that's why I get little confused. Sorry.
This graph is showing diversity in different families. Okay, and you can presume that nearly 6 to 7% of diversity is of all the faunal groups is present in India. Like 1 lakh species of animals are present. You can see, and out of 18 lakh species so far described from the world. Okay. Then these are some pioneer works from Codeopra, and I welcome anybody from the webinar, any student, any researcher, any PhD student to here I am now as in charge of Coleoptera section. You can send me specimens and photographs through email or WhatsApp. We will try to identify your specimens as far as possible. Though it is not possible to identify all the specimens, all the photographs, but we can help you in identification. If your MSc, some project is there or some PhD is there you are doing on beetles, and some data you need or anything, we can help you in those things, even for your taxonomic work or studies. Okay. Then other groups, that is Lacewix, these are very important group. You must have seen Chrysoperla sirena, that is important predator of different aphids. 342 species of this order is known. Okay, then caddisfly, I previously told it is one of the important group. One of my friend, Dr. Manpreet, is here in Geological Survey of India. Dr. Manpreet Pandher is working on this caddisfly, and these are also present in aquatic environment and susceptible to environmental change. Okay, then butterflies and moths. Everybody knows there are on the left side there are two photographs. One is a below one is a butterfly, the upper one is a moth. So there are 12,500 species of moths and butterflies. These species inventory ZSI actually maintains. You will be surprised how I know this number of species are known from India or like that. We actually in ZSI we catalog the species. It is our mandate. It is our objective to catalog and inventorize the number of species of every group present in our country. Okay, and everybody knows about these groups, so I will not be telling much. And especially we attract towards butterflies too much, but butterfly only 1,500 species are known, and moths we do not attract too much. But this is very important group, and they are and in ZSI we are also assessing the pollination of. If Dr. Navneet Gill is here, he is senior scientist. He is assessing the pollination potential of moths in Himalayan region. These are certain butterflies from Himalayan region. If you see this, I am showing this is a light trap. We are using a light, and you see the moth diversity. There will be 50-60 species of moths in one sheet. It is a one, you can say 1 feet area. They are moths, and it is from Nicobar Island. We get. Then some Diptera, the 7,220 species are known. Like you can see mosquitoes, then flies and sarcophagids, many horseflies. You will be surprised to know that there are more than 400 species of mosquitoes from our country. And what we know generally is Anopheles, Anopheles and Culex. Very few species we know. Okay, then fleas, they are a very important group. 46 species of this group are known from India. Okay, this very well worked group in India, fleas. Then scorpion fly is very rare to sight in the field and very rare to collect. Only 23 species of this group is known from India. Okay, then comes to the. But we have somehow some idea we have about how insects look like, what are different types of insects, what are the names associated. Now comes how to study them. For studying in taxonomy, we first collect them, then collect them by different methods, by applying light trap, baited trap, sweeping, different methods we apply. Then after collection, the preservation of those specimens are very important in the laboratory, because then they will be a reference collection for years. Like in ZSI, we have collection nearly 200 years specimens are there, and they will be remain here for thousands of years till ZSI will be here. Those specimens will be here. Then we study them, identify them through monographs, field guides, and keys. But how this survey actually start? First, for going particular, like suppose I want to study beetles and certain area like suppose Arunachal Pradesh, what requisite we require? What information? First is you need to know about the geography of that area, climate condition, vegetation of that area, so that you will be what group of insect you want to collect. So based on that, you need to know about that area something. About their climatic condition, when to go. This will help you in the in planning of your survey, and the permission. Before going to the field in any protected area, any state, take proper permission from the state government department, state forest department, and and have one GPS also. Draw a map in Google Map or something, whatever area, and and then select your area. Like I want to work on dung beetles, then we cannot go in an area where herbivores are not there. There we will find no dung beetles. Okay, so we need to know about something about that particular area, their vegetation, and all those things. Then comes the permission. Then comes the sampling method. How will you collect all those specimens? Whether you will hand pick, light trap, you will make light trap, pitfall trap, soil emergent trap, bunnies funnel trap. Then comes when you collect them, you kill them in benzene or anything. Then proper labeling is required. Like if you collect on 16th July today, you need to write which place you have collected those samples, which date you have collected the samples, what is the latitude, longitude, and altitude of that particular area. And then comes the who has collected and habitat, like from some leaf you have collected, from roots you have collected, and what means habitat, whatever information you know, and take a picture from your mobile also. Sometimes it helps a lot, baseline information. Then the preservation comes. In the field, preservation is different, and then you come to laboratory and preserve them. In the field, you keep them in envelopes or alcohol vials and label them. Then you come to the laboratory and stretch them. Then these are certain habitats. In green vegetation, you will find Lepidoptera, Coleoptera, Hymenoptera, Hemiptera, and under stones, bark, crevices of trees, decaying logs, you will find Protura, Dermaptera, Embryoptera, Sophoptera, and Coleoptera. Coleoptera you will find in every habitat. That drains, go downs, domestic dark. You should know what you want to collect. If you want to collect cockroaches, you go to drains, go downs, domestic dark, and food funnel. But in some of the very good cockroach species are present in forest. Then if you want to search for collembolans, humus soil you will be needing. Then diplurans also in soil. Then isopterans, logs, mud mounds, under stones. Then based on like water bodies, if you want to select for aquatic, I previously told Ephemeroptera, Odonata, Ephemeroptera, mayflies, dragonflies, damselflies, caddisflies, stoneflies, aquatic bugs and aquatic beetles. You will find in water bodies, mostly at these shallow water. Then if you want to find the Theraptera and Siphonoptera, you have to go for this skin of mammals and birds. Okay, then parasitic, another, etc. Then what are the things you will require in the field? One is for collection of insects, I suggest that mercury vapor lamp is most important. It is 125 watts mercury vapor lamp, Philips mostly, and Surya also now it is stopped. You can very rare you will find any old shop, electric shop, then you will get it. Otherwise, new electric shops, they are not having this, and very rare. You must have this when you go in the field for collecting the specimen. Then second chemical what you need, one is benzene and other one is 100% alcohol, absolute alcohol. 70% alcohol will also work. Benzene and ethyl acetate both you can have. For butterflies and moths, You can use ethyl acetate. For beetles, hard insects, you can use benzene. Then the next thing you need, entomological pins. The entomological pins come from 0 to 5 sizes. Entomological pins are rust free. And then insect envelopes of different sizes. Then you need a GPS. Then you need an environmental meter. Then you use spade. Then forceps, scissors, and vials of different sizes you need. Then this is a baited trap for collecting ground beetles and dung beetles. And this trap we applied in Great Nicobar Island. And recently we published a new species from this collection, Clyster. Galathea National Park is there in Great Nicobar Island, and we named it Clyster galatheensis. Okay, and all the things you need for your protection. Insect nets are required, and sometimes these big sheets are required if it's heavily raining. And you see these vials. These are killing jars. Okay, larger size, smaller size, very smaller size. Depending upon the size of insects, you need to have a series of killing jars so that you can collect the specimen based on the size and size of the insect and the jar. Okay, then how to collect them? You can, for butterflies and moth. For butterflies you need a sweeping net, and through that butterfly net you can also sweep some smaller insects like beetles from grasses. Then aquatic net is required for collecting aquatic hemipterans, bugs, and ephemeropterans. Then pitfall trap for dung beetle. Then beating net. You will beat the trees, then you will find this light trap, aquatic net, common insect, any aspirator for taking the insects. Okay. Then for stretching you will need these spreading boards. Insects are spread as they look in the field. Means we need to keep the specimens in the museum as they look in the field. Okay. Then these spreading boards are required for, especially for butterflies and moths. And these are different sizes of entomological pins. Then pinning is most important, especially in the case of terrestrial specimens. In case of aquatic insects, we keep them in alcohol. Like aquatic insects, they mostly live in aquatic life, and in museum also we put them in aquatic, like 70% alcohol, like trichopterans, ephemeropterans, and these dragonflies, themselves flies. We keep them in aquatic 70% alcohol. But some dragonflies we pin also. These are and for pinning, inserting the pin, entomological pin, there is certain position you need to know. In Hymenoptera, slightly right side, and in Hemiptera at scutellum, but the pin should be at right angle. Okay, and 1/3 of the pin should be above, and 2/3 of the pin should be below the insect. Okay. Then this is the information you need for a particular group. Like we went to Nicobar Island. This is a field label. It is having a group of what group of insect, then where it was collected, whether it is dry condition, which district, which latitude, which particular locality. It will actually help to analyze your results after you come to the laboratory. This thing is very important. And then 2 permanent labels are required. You just short it down, and the one is locality label, the identification label, who has identified the specimen, what is the registration number, that is required. Okay. Then this is the position. You can see pinning. How to that 1/3 very small that you can pick is upside, and 2/3, because here you will require many pins, many labels. Locality label will be there. If it is a new species, the type label will be there, and identification label is there. Then you have certain DNA you want to extract, and that label will be there. At least 6-7 labels will be there. Okay. Then mounting is also done, where insects are very small. Then we put them on a card and glue them by a water soluble glue. That way we do. Okay. Then these are the certain boxes we have to arrange the specimen, like jewelry, very fine space and everything. These are certain insect cabinets we put, the insect drawers we put, and I welcome all of you whoever in Calcutta, if the situation gets normalized after 2-3 months or something, anytime you are welcome to visit Zoological Survey of India with the permission of your principal madam, and we will be happy to show you all the insect collection in different section, and you will know how to study them and what are their insects. Okay, then after collection, the main thing is study.
Thanks a lot, sir.
And this is actually like a microscope. We do the photography of the insects. Lots of things, and this is after the collection. We study the higher resolution, the characters and everything. Okay. Now we need very good photographs for publishing our studies, and drawing also we do. You see the right side, there is a drawing tube. Many of the groups photographs are not very important. Photographs do not come very good, but you need to have draw the male genitalia. You need to draw for comparing with the previously known species. Okay, this is a Nicobar Island. We were collecting in 2018. Okay, this trap we put in 2018. You see, it is nearly 2 o'clock at night. We are putting, and there was rain. We use umbrella for collecting. Okay, and that bulbs we use 125 watt mercury bulbs was broken, and we did not. We took 4 bulbs, 6 bulbs with us, but that was broken. We need to purchase these CFLs. But you see, in the CFL itself, there are huge diversity. In Nicobar is a treasure. These are the vials, different vials. You see, for butterflies, there is a different vial. For alcoholic vial is there. There must be Ephemeroptera is there. You put the specimen in the specific vial, and continuously you clean them so that your specimen get not infected. We care less about ourselves in the field, but more about insects in the field. This is, you see, from this trap only. This trap only we collected these specimens, and we sorted out in species groups. In the field itself, you can see morpho species. You can differentiate. How many species? I think there will be nearly 55 to 60 species in this tray only. Okay, there are hawk moths are there. Many groups are there. And this photograph, and always we nowadays have our cameras in our mobiles. We are having smartphones. Whenever you go to the field, I suggest all of you take the photographs. If you do not have DSLR, no problem. Take the photograph through your mobile camera. That is the most important thing. Do not go for higher price this DSLR camera and all these things. It is not required. This is a mobile photograph. Means through mobile photography, you take the photograph first, then you collect the specimen. That is the most important thing. Then identification, I will suggest it needs certain expertise. It gives you after 2-3 years of training in taxonomy, then you can identify the variation in the species, genera, subfamily, family, all those things to understand the characters, level of characters and weightage of characters, which character is important at order level, which character is important at family level, which character is important at generic level in identification. Then you can also help, you can also take the help of expertise now through email. You can contact the expertise, but for specimens, you can go for ZSI, IARI, FRI, and NBIR in Bangalore. For insects, I am telling. For other groups, other fishes, different group, mammalia, different. Mammalia, there is very good field guide, no need to. And collect very less number. I will say that do not collect all the specimens. Collect few, means 4 or 5 specimens of particular sample, and deposit them in depository, like ZSI and BSI plants are there, IRI, so that that will be used for future generation. Okay, then you can make publications, taxonomic publication like monographs. Monographs are the most important publication in taxonomy. That took nearly 20-30 years of work. After that, you can publish a monograph on particular group of insects, a particular genus, particular family. Then revision are a second important publication in taxonomy. I'll suggest you all, there is one book that is called An Introduction to Taxonomy by Dr. T.C. Narendran. It is available in faunaofindia.nic.in website. You can go and read. Very small book, 60, 70 pages book. You can read in your, you can download in your mobile and read it. Very important book for the taxonomic science. Revisionary work, then revisionary means genus level revision, subfamily level. Actually, this gives the identification of particular. Then reviews, atlas. Atlas is actually photographic, not a textual part. Atlas includes only photographic, like butterflies of Calcutta. No need of any text, only the photographs only. Then description of new taxa, like new species, new genus, new sub. And then checklists are important, like state checklist, biogeographic area checklist, regional checklist, global checklist, catalogs, and history. These are publication. Thank you for all, and I welcome any question from the participants.
Thank you, sir. Thank you
Monica
for
ma'am is
your
there?
interesting and informative lecture. Now we'll go
Thank you, ma'am.
ahead
Thank you.
and take some time for questions. Ononno,
Okay.
if there be any question.
So it will be better if you can type your question in the chat box. I think one question is there already from Shahana Mukherjee. So she asked that about if there is any habitat preference for insects, particular which type of habitats are more preferable for insects.
Yes,
If I'm not wrong, I think.
Yes, I will give you. There is one slide. Like if you want to go for soil insects, okay. Then then you need to study the ecology of that particular group, like collembolans. Which particular habitat is having like soil. Collembola will be present. I showed one slide. Aquatic insects. You will find trichopterans, ephemeropterans, and all those. There are two types of research you can do in these insects. One is habitat specific. One is taxa specific. Habitat specific means aquatic insects, suppose, and taxa specific means like beetles or butterflies. Then you need to have a different view. If you want to aquatic insects, you will collect different group: Ephemeroptera, Trichoptera, aquatic bugs, and aquatic Coleoptera. If you want to study particular beetle like Curculionid beetle or daddy beetle, you need to go for that thing. And there is one question from Saurav Ghosh. He is asking that day by day the diversity of the insects become decreased due to anthropogenic activities. So how to conserve and increase in insect biodiversity in such I suggest this is very important question he has asked actually. This anthropogenic activities, what mostly we do, we study the impact of these pollution, habitat destruction in case of larger animals like tigers, mammals, reptiles. We do not bother much about insects. So we need to look into that and how to conserve them. Actually, we need to do our means human beings need to restrict ourselves for these anthropogenic developments and all those things. We need to do something, and if we will stop development in unsustainable way, then definitely the insect population will increase.
Okay, another question is from Neha Roy. I think she belongs to our department from Sirampur Girls College. Why Hemiptera is called true bugs?
Hemiptera is not called true bugs. The hemipterans are called bugs. Heteropteran are called true bugs. True bugs means bugs actually is a term for two, three groups of insects. Okay, Coleoptera, also sometimes called as bugs, but in only hemipterans are bugs.
Question is from Arunashai. So, how to differentiate a grasshopper and locust?
Yes, very good question. Very good question by actually these grasshoppers, all the locusts are grasshoppers, we can say, but all the locusts are grasshopper, but all the actually it is a transformation from the solitary to gregarious phase. Means this because of drought and drought condition and environmental conditions, simple solitary stage may become gregarious. There is one species that is called Cysticerca gregaria. It is the species name is gregaria. That when the situation are normal and the temperature is normal, it will remain like simple like grasshopper. It will behave. But when the situation becomes drought and extreme hot, they start they start producing in huge number of individuals, and for their preserving their species, and start consuming, and that is called gregarious period. That is the locust in environment that through environmental and food scarcity, they transform from grasshopper to a locust.
So there is another next question from Deepak Kumar. So I think we both know him. So pollination is essential. We all know how we can ensure the availability of insects for you while we use insecticide and all.
Deepak Kumar, hello Deepak. How are Deepak? Pollination is very right. Pollination is essential, but we actually in case of insects we have studied. We use organochlorinated, then we use organophosphates, then we in case of insecticide now we are using pyrethroids. Now we are moving toward essential oils and all these things. We are switching. Means we are using insecticides because of food security for controlling the pest species. Actually, many of the pest species in the field are very less, and because of them, many of the non-pest species are also killed, and that is affecting our bees, especially bees. And it is well known fact that use of insecticides are reducing the population of bees in the field, and because of this the food security will undoubtedly. But there are certain things like biological control we can go, or anything, so that the less use of insecticide will be there. But what is this biological control use of trichogramma and these predators are good for our agriculture? But these questions are required to be answered, because we need to feed a huge population also. That's why the sustainable use of insecticide, we can say, we can do.
I think that's why that biological controls that part will come here.
No, biological control is important, but my biological control, but how effective it is in controlling the pest population, means that is the question. We need to change. Actually, we in agriculture we are mostly like in Uttar Pradesh. We are only sowing sugarcane. We are not showing other crops. Then what happens? The insects actually change their feeding habit. And many of the common insects are becoming pests nowadays. Means the pest status is changing because of these monocropping also. So we need to use pulses also. Interchangeable crops are not there. Lots of issues are there. So these are hampering the insect population, insects into pests.
Okay. Actually, there is another question from Shomnath Mondol. I think in botany there is a problem of termite infestation into fungal cultures in PDA. Is there any way to inhibit that without causing harm to termite diversity? That's a very clear.
Of termite infestation into fungal culture. I didn't get actually PDA.
Can you please explain about the PDA? We are not really. Okay, we'll go to the next question till the explanation of PDA will come. So, sir, how can we study life cycle of any particular insect? From Shivani Gupta.
Very nice question. It is very important. I suggest taxonomy is one part and studying life cycle is the other part. Studying life cycle of a particular insect is very important, and for very few insects we know the life cycle. For like suppose if you are dealing with a particular butterfly, first thing you need to know whether what type of eating it does. Like you need to observe it is a monophagous, oligophagous, or polyphagous. If the particular insect is having food is most important. That if it is particularly feeding a particular plant, then you need to know that, and you need to provide that particular plant for that. First thing, like you can start with Discus. There is one particular group of bug. You bring one male and two females and put it in a container with the soil inside the container. Below is the soil, because most of the insects lay eggs in the soil. Okay, then for 10 and then you can use some cloth to cover it, and you keep it in temperature. Better to keep it in incubator. We did do it in incubator, but for study you can put it in there. Okay, then the eggs will be there. Then after hatchling you can provide the food. As I told, if it is a polyphagous, you can provide anything, especially castor. Castor leaves are mostly used for Lepidopterus insects and other food. Then this okra and all those things in case of you can study them. It's very easy and very interesting life cycle. Okay. Then you can start feeding them and note down every stage. Like after how many days they lay eggs, and what stage particular group is. First nymphal stage, how many days? Second nymphal stage, how many days? Third nymphal stage, how many days? How many nymphs have come? How many legs are there? Eggs are there? Everything you note down in your booklet, and you will get very good paper. You can produce out of life cycle of any group of insect if it is not published, and it will be published in very good scientific journal.
Okay, so last question for today. I think Somnath Mondal has provided that explanation for potato dextrose agar, that is the PDA. So I think he is culturing some fungus on culture media, that is the potato dextrose agar. So in there some infection from termite is going on, and he is interested to know how to check that infection without altering the diversity of termite. So it's really good question.
He is doing it in the lab, then I don't think termites are required. They will live in natural environment. No problem. You can remove them if it is doing in lab. No need to grow termites in the lab. Usually,
I think every time we try to remove it using, I think, alcohol or something to wash all the discs and other things.
Can have alcohol can remove any insect, but need not to keep them. Actually, if you are doing certain research on something that you need to other things you need to remove. Okay, that thing will be a contamination. Termites are contamination to his.
Yeah. So if we just want to remove the termite from our lab, I mean from inside a home, so we doesn't need to alter the diversity at the field. So field diversity
no. In the laboratory, no, it will not alter. Actually, one insecticide is there, that is chlorpyrifos. That can be used for this removing the especially for termites. You can see I do not have
exactly. Okay.
Chlorpyrifos. And this one question is very important. Usually insects are killed.
Okay.
Suhana Mukherjee. Usually insects are killed or preserved for identification. Is there any way to identify?
She is from an environmental biologist from NEHU actually.
Very nice question she has asked. I would like to answer it. First thing is you can identify what purpose you need the identification. First thing, killing insect killing insect is not harm to the biodiversity. First thing. Why? Because I previously I explained to you that insects are having life cycle and egg, larva, pupa, and adult. In any, if you kill an adult, it will remain in the egg stage or larva stage at any time. You are not going to, but do not kill deliberately insects. If you want to study insects, you can collect them in very limited numbers, not in large numbers. Second thing, you can identify the specimens through insects. But the problem is you can identify butterflies very easily. Very good field guides are there. You can identify certain moths, certain beetles, which are very well studied. But all group of insects you cannot identify through photographs because it needs expertise to identify a particular species. We need to compare the genitalia. We need to sometimes go to the SEM level, scanning electron microscopy level, for identification of particular species. But certain groups, odonates can be identified, butterfly can be identified, certain group of moths can be identified, certain beetles can be identified. But I don't think that ephemeropterans and smaller insects can be identified through photographs. And we need to preserve them and study them under microscope for identifying. There is one question by Nabeshri Yadavi: in field, shall we collect the butterflies or not? First thing, for collection, I think everybody need not to collect everything. First thing, like you are working under one supervisor, you first take the help of your supervisor. If you are doing PhD or MSc or BSc, whatever you are doing, take the help of your teachers, supervisors for collecting. Just do not collect them for fun. Because there are strict laws for collection, you need to work under your supervisor or teacher so that he or she may guide you how to properly collect them. And do not collect them for the sake of fun. Collect them, you can collect them, but you need to deposit them inside in any national depository after your research. But be in contact with your supervisor or teacher. Work means that will help you in doing this. Very important question. And first, the important thing, do not collect anything inside any protected area. We have national parks, wildlife sanctuaries, conservation area, community reserve. It is a crime to collect anything inside a protected area. Killing a butterfly inside a protected area is as equal crime as killing a tiger. Okay. As we know that insects are very much important for collection of honey. So is there any particular method to preserve those kind of insects? Insects are very much important for collection of only actually he is actually Madhushri Haldar is wanting to ask about the bees. Or I think bees, not only honey is provided by certain bees, Apis mellifera, Apis sirena, and Apis mellifera is most important bee for collecting honey. The only way is the sustainable use of pesticide. You can conserve those bees, and bee industry is very good. Like in West Bengal, it is very good to have. Okay, Ananya.
Hello.
Thank you
for a very much informative presentation in our webinar.
Thank you, ma'am. Thank you, and thank you. Especially, I would like to thank Principal Madam and Dr. Ananya. I would like to thank Madam Dr. Soma Roy, Principal Shirampur Girls College. Respected Madam, thank you very much for giving me the opportunity to interact with the participants and through webinar. And thank you so much, Madam. And I would like to thank Madam Dr. Sayanti Mukhopadhyay, Talukdar Madam. Thank you very much, Madam, for your and especially for your talk, which you gave very informative. Dr. Ananya, my friend, and joint convener of the program, Dr. Mayur Kashi Nag. Thank you very much, madam.
Now, Anurna Ali, Dr. Anurna Ali. Hello.
Hello.
Yes. Who will give the vote of thanks?
Okay. Hello. Yes. Hello. Am I audible?
Yes.
Yeah, you are on.
Okay. So currently, I will provide that vote of thanks.
Hello.
Yes. Am I audible?
Yes.
Okay. Just give me 2 minutes. Okay. So I'm audible right to everyone, right?
Yes.
Okay. So I would like to thank UGC and Calcutta University, Srirampur Girls College, and the governing body of the college for allowing us to have this opportunity to arranging such a great academic affair for the from the college. On behalf of college, I would like to thank Dr. Shoma Roy Jana, principal, and also the Secretary of Teachers Council, Bimal Mondol, as a GB members. Also, I would like to thank the Zoological Survey of India and Dr. Devanshu Gupta for being our guest and honorable speaker person, and his informative speaking has really enriched, I'm sure, our participants and as well as me. And also, I would like to thank IQSC team and the coordinator of IQSC, Dr. Shanti Mukherjee Talukdar, for continuous support and enthusiastic speeches that boost the spirit of the organizers and participants. Also, I would like to thank the whole webinar committee, Dr. Shampa Sarkar, Dr. Samuel Daripa, Dr. Uttam Haldar, and Dr. Sandeep Majumdar, and many in behind them, for their tireless effort of these people has made this event a success during these tough days. And also, I am highly thankful to the all faculties of all stream for their continuous support. And there is we are actually in a continuous method of webinar forming and arranging and attending. So this gives actually really good mental support to us. So also I would like to thank Dr. Muyurakshi Nag for hosting the whole program with such elegance, and she is also the joint convener of this program. And also there are some people whose name need to be mentioned, particularly like Devkanti Das and Sandeep Majumdar for their continuous role in setting all the required technical background. Without them, I myself won't be able to build all this thing, and I am personally owe this to them. And also Naumita Ghoshnath, madam, for her awesome role in draw and design the posters for most, if not all, the webinar. Also the participants, they need to be mentioned for their huge warm response. We didn't. Frankly, have not thought about that much warm response. So I'm really flattered of having this response. Without them, all this webinar won't have much meaning, as it has been designed mostly for their interest only. And we owe them their valuable time and expecting that it has been fruitful. And thanks all. Okay.
Now,
Moiraki ma'am is there?
Okay. Thank you all the participants for making the event successful. Now I request our principal madam to announce the end of the webinar.
now we come to the end of the webinar, and I thank all of you to join in this seminar. Let me close the webinar. Thank you.
Okay. Bye and good day.